Today we will study about beta-lactamase inhibitors, in which we will see the classification of beta-lactamase inhibitors, then mode of action of beta-lactamase inhibitors, and the study of individual compounds used as beta-lactamase inhibitors. Firstly, what is beta-lactamase inhibitors? The beta-lactamase inhibitors such as sulbactam and tazobactam and natural occurring beta-lactams such as thiamycin inhibit both the beta-lactamase and interact with penicillin binding protein present in the bacterial cell wall. The beta-lactamase inhibitors are given along with beta-lactamase sensitive penicillin so that they competitively bind to the enzyme and protect the penicillin from destruction. Beta lactamase inhibitors are called as suicidal substrate. Now we will see the classification of beta lactamase inhibitors. There are two classes of beta lactamase inhibitors: class 1 inhibitors and class 2 inhibitors. In class 1 inhibitors, they have a heteroatom at position 1. Its examples are clavulanic acid and sulbactam. The structure of clavulanic acid and sulbactam is given on the screen. Now, class 2 inhibitors. They do not have a heteroatom at position 1, and its example is carbapenem, where there is thiamycin. The structure of thiamycin is given on the screen. Now, we will see the mode of action of beta lactamase inhibitor. Inactivation of beta-lactamase is done by mechanism-based inhibitors, which act by reacting with the enzyme in the same way as that of the substrate. An acid enzyme intermediate is formed by the reaction of the beta-lactam with an active side serine hydroxyl group of enzyme. For normal substrate like penicillin, the acyl enzyme intermediate readily undergoes hydrolysis, destroying the substrate and freeing the enzyme to attack more substrate. For mechanism based inhibitors, the acyl enzyme intermediate is diverted by tautomerism to a more stable amine that undergo hydrolysis more slowly to eventually free the enzyme. Because these inhibitors are also substrates for the enzyme that they inactivate, they are sometimes called as suicidal substrate. Now we will study about the individual compounds which are used as beta-lactamase inhibitors. Firstly, we will see clavulanate potassium. The structure of clavulanate potassium is given on the screen. Clavulanic acid is isolated from Streptomyces clavulgaris. It is 1-oxypenem. It lacks 6 acyl amino side chain but possesses 2-hydroxyethylidene moiety at carbon second. It has very weak antibacterial activity. It is a potent inhibitor of Staphylococcus aureus beta-lactamase and plasmid-mediated beta-lactamase produced by gram-negative bacilli. Combination with amoxicillin and potassium salt of clavulanic acid, it gives augmentin, which is intended for the treatment of skin, respiratory, ear, and urinary tract infection caused by beta-lactamase producing bacterial strain, where the bioavailability of both are the same. It is effective against strains which are resistant to amoxicillin alone. It is also stable to acid. Combination of potassium clavulate and ticacillin sodium, that is extended spectrum penicillin, is recommended for streptissima, lower respiratory tract infection, urinary tract infection, bone and joint infection, skin and structure infections, caused by beta lactamase producing strains of Staphylococcus aureus, Klebsiella, E. coli, Pseudomonas aeruginosa, and other Pseudomonas spp, Cytobacter spp, Enterobacter spp, and Serratia marginum, etc. Now, dicarcylin disodium. The structure of dicarcylin disodium is given on the screen. It is beta carboxyl 3 thiolene penicillin. Now we will see the class 2 inhibitors, which is carbapenem, class of highly effective antibiotic agents commonly used for treatment of severe or high risk bacterial infection. This class of antibiotic is usually reserved for known or suspected multi-drug resistant bacterial infection. It is broadest coverage of bacterial activity, including gram-positive and gram-negative, that is, especially drug-resistant species, and aerobic coverage, cover MSSA, Enterococcus, Streptococcus spp, and the drug of choice for ESBL infection, where ESBL is the producing bacteria that can't be killed by most of the antibiotics. Now, in carbapenems, we will see thiamycin. Thiamycin is first isolated from Streptomyces cattleya. Only two structural features of thiamycin are shared with penicillin and cephalosporin. It has a fused bicyclic ring system containing a beta-lactam and an equivalently attached 3-carboxyl group. The presence of double bond between carbon second and carbon third in the bicyclic structure creates a considerable ring strain and increases the reactivity of beta-lactam to ring opening reaction. It has 1-hydroxyethyl GP at 6th position, where not the acyl amino side chain, and this is oriented to the beta ring. It has broad spectrum antibacterial properties, which is active against gram positive and gram negative bacteria and resistant to activation of most beta lactamase. That could be because of hydroxyl ethyl side chain. It is more susceptible to acid and alkaline hydrolysis cause the strain of nature of the fused ring system. It is stable between the pH 6 to 7. Now we will see imipenem. It has very stable to most beta lactamase. It is an inhibitor of beta lactamase from certain gram positive and gram negative bacteria resistant to other beta lactam antibiotic. It is used for the treatment of wide variety of bacterial infection of the skin and tissues, lower respiratory tract, bone and joints, and genitourinary tract infection. They are also used for streptissima and enterocarditis caused by beta lactamase producing strain. Thank you.